Good afternoon, everyone. So on behalf of Professor Jesus Banales, he couldn't be here today. So I'm presenting the group he's leading at the Biodonostia Health Research Institute, the liver diseases group. So as Patricia previously mentioned, we are also located in the Basque Country, so in the north of Spain, 1 hour away from Patricia's group. So we are even closer to the French border, but we are still in Spain. So the liver disease group, as I mentioned, is led by Professor Jesus Banales. It is a multidisciplinary group with basic and translational researchers. We are basically two, apart from Jesus, we are two principal investigators, Maria Jesus Perugori and myself. And then we have 4 postdocs in the lab and 5 PhD students. Apart from the basic and translational researchers, we also have very close collaboration with clinicians in our hospital, since we are located in the Donostia University Hospital. So we have different clinicians in our group. We have hepatologists, surgeons, oncologists, radiologists, and also the head of the clinical trials of our institute is also a member of our group. So as lines of research in the group, we are mostly interested in studying liver pathobiology. So regarding the biology of the liver, we study a lot of bile acid metabolism and how microRNAs, extracellular vesicles can contribute to the maintenance of the biliary, mostly biliary phenotype, and also aim to characterize the biliary 3. Going to the disease mechanism. So one of the lines of research of the group is mostly related with the study of chronic liver injury and the progression toward liver cancer. So in this research, in this line, Maria Jesus Perugori, or Machus, as you most of you know her, she's studying mostly innate immunity. She's focusing her research on TREM2 and marker receptors, and she's studying the mechanisms of inflammation and fibrogenesis, mostly focusing on the non-parenchymal cells in the liver, such as Kupffer cells or hepatic stellate cells, and she's creating a new line of research in immuno-oncology. Apart from chronic liver injury, we are also studying non-alcoholic fatty liver disease. We are touching the diagnostic part, so we are looking for new non-invasive diagnostic tools to diagnose NAFLD and predict disease progression. And in this case, as an example, we are focusing on studying metabolomics. We study also genetics and epigenetics in the NAFLD field, and we are now studying the progression of NAFLD towards liver cancer. And one of the most, the biggest core groups studies in our group is the study of cholangiopathy, so the diseases that affect the biliary tree. So in this sense, we have lines of research in autoimmune liver diseases such as PBC or PSC, in which you study not only the pathogenesis, but also try to identify novel diagnostic biomarkers. We also study polycystic liver diseases, and the major arm of the cholangiopathy study is cholangiocarcinoma, in which we study the mechanisms of pathogenesis, try to identify novel diagnostic and prognostic tools, and also are interested in the study of chemoresistance. So Professor Jesus Banales leads all these lines of research, and then all the PIs and postdocs are leading specific lines. So in our case, Machus is leading the progression of chronic liver disease line, and also leading the cholangiocarcinoma immuno-oncology research. Myself, I'm leading the part of the cholangiocarcinoma development and progression, and mostly now starting a line of research on NAFLD-associated liver cancer. We have Dr. Laura Izquierdo. She is a postdoc in the group that is the one in charge in the clinical study of cholangiocarcinoma, and she is co-leading the European Cholangiocarcinoma Registry with Professor Jesus Banales. Then we have Dr. Ainhoa Lapid. She goes with the part of biomarkers, and she's leading all the research in liquid biopsy and extracellular vesicles in the group. And then we have Dr. Francisco Javier Caballero, which is studying mostly the part on cholestasis and new therapeutic targets. So, and techniques and methodologies that we are mostly use on daily routine in the group. So we do a lot of in vitro experiments, so cell culture. We do loss and gain of function experiments. We try to delete genes with CRISPR, with shRNAs. We try to overexpress genes of expression using lentivirus. Then we characterize the cells by flow cytometry, such as to measure proliferation, cell death, oxidative stress, try to evaluate mitochondrial bioenergetics, do some assays to check migration, invasion, and all the techniques that are related with that. But we also do a lot of in vivo stuff. We had several animal models of liver injury, such as models of CCL4 with thiocetamide, models of cholestasis. We also have and conduct models of NAFLD using diets. And we have several animal models of liver cancer, mostly of CCA and HCC, using carcinogenic genetic models, xenografts, allografts, and so on. And we also do a lot of experiments with samples from patients. So since we are in the hospital, we have access to a lot of samples from patients. We have from tissue to biofluids that include serum, plasma, urine, sometimes feces. And we also have the registry of all these patients with the clinical information. And we are creating databases and leading patient registries. So this is one image of our group. As you can see, we have a beach nearby. The city is really, really beautiful. And if you have any questions to our group, you have here the contact people that you can approach to get more information. And thank you very much.